how does the age of horses correlate to their gut microbiome? Our hypothesis was that the older the horse is, the more diverse their microbiome will be. Our rationale for this is that an older horse has been exposed to many different environments. So our four samples of horses, their breed is thoroughbred. Their ages are 7, 14, 4, 9 months, and 14. Um, they all had good health. So for our materials and methods, we first isolated the DNA with the QIAGEN kit to prevent the enzymes from breaking down and to wash the impurities. We then used the Zymo kit to concentrate the DNA to work with a better concentrated samples for more consistency. We then used UV spectrophotometry to determine concentration and find impurities at ranges 260 to 280 nanometers. Then we used the amplicon PCR to amplify the 16S ribosomal RNA gene to prepare the primers for synthesizing DNA. We raised the temperature to 72 degrees Celsius to synthesize the DNA. Then we used the agarose gel electrophoresis to check our PCR products. After that, we used the Asian Core and Pure Kit to remove the primers and any other reagents. Next, we did the index PCR to add the tags and identify which sample was which. We did 8 samples, all at 72 degrees Celsius. After that, we did another PCR cleanup. And we quantified our data again using the fluorimetry. We use this because it is more sensitive than the UV spectrophotometry and could distinguish between DNA, RNA, and protein. Lastly, we did DNA sequencing using the alumna core. Our instructor used this to determine the nucleotide sequence of our samples over the Thanksgiving break. So for our results, our concentrations respectively were 137.6 for sample 331, 110.3 for sample 361, 20.6 for 408, and 57.5 for 885. And this is all in nanograms per microliter. Our ratios for the UV spectrophotometry for 331, it was 1.76, 361, it was 1.82, 408, 1.79, and 885, 1.58. And here is our gel electrophoresis kit here. You can see here that for our negative, there really isn't much light here shown. And for our positive, there is much light shown. You can see that for all 4 of our samples, you see that there is DNA. So for our number, all of our reads were successful. Everything resulted in 100% after using the alumna core. And so here's our analysis for those analyses. So at the kingdom level, all of our samples had read for bacteria at 100%. For the order level, you can see here that there has been more diversity. You see in this sample is actually, you can see for this classification, the enterobacterials, you see that our sample 331 had 100% and our positive control had near 100%, while you see as the rest of the classifications, it really ranges. And for the genus level, everything is sort of more leveled out, but everything really just, it just varies, so it's very diverse here as well. There were also 2 more classifications. One was the unclassified. The unclassified section did not match any known organisms. And then there was also another classification called other, which is just the bacteria, the classifications that were present, but then had a less than 3.5% total reads. So overall, our concentration was high enough and our reads were 100%. All of our samples during gel electrophoresis were visible, so the reaction worked as expected. For our positive control, our reads were we had 8,432 reads, meaning 100%. We found that bacteria from the skin was also found in the analysis, which means that contamination may have occurred. So our conclusion, based on these results, we have determined that no, old age did not correlate to having more bacteria in the gut microbiome.